Okay, 116 students, here's your promised extra summary for the end of chapter 23. It was a bit rushed today, so we need to do better justice. Passive transport is always when substances are moving across the membrane from high to low concentration areas. These are depicted on the left part of this diagram, where you see the things are going through the membrane or through a pore, but they're going in the direction from high to low concentration, and it happens one of two ways. Either it's going to happen in simple diffusion case, where only small things can get through, like nonpolar molecules such as oxygen and CO2 across the membrane without the benefit of a protein, or if it's got to have something else helping it along, it can be facilitated diffusion, where there's a protein involved to allow the molecules to go from high concentration to low concentration through some kind of a protein channel or a gate. They have different features that can allow molecules to get across the membrane. The one in the middle that suggests it's called facilitated diffusion is one that shows that's opening like a gate, open on one side for a minute and then closing and then opening on the opposite side to transport the molecule. These are both passive in the sense that they go from high to low concentration, and molecules that are bigger than a nonpolar small thing like oxygen or CO2 are going to require facilitation by a protein. The third type of mechanism here is called active transport, because you can only make things go across the membrane in the sense of active transport with the benefit of energy added. The energy added is going to have to be something like ATP, but active transport will take things that go from a low concentration area across the membrane to push them all to a high concentration area. It's like the sodium-potassium pump that I tried to describe briefly today, but it does require energy, and it ends up pushing things all one way across the membrane. There's another diagram for that in a minute. So only with the benefit of energy can active transport take place. You push things from a low to a higher concentration, and it's how concentration gradients are maintained inside and outside of cells, which is what is necessary for ordinary nerve impulses to take place. The sodium and potassium gradient is such that you push, the pump is going to push all the sodium to the outside of the cell and the potassium to the inside of the cell. Not that you need to memorize which is which. And the reversal of that occurs with membrane or nerve transmission, and then the pump has to restore that gradient to put potassium on opposite side from the sodium. ATP required to do that. So another diagram for the active transport with the sodium-potassium pump is here from your textbook, of course. That's the wrong figure number. It's in chapter 23 now. But it shows you where the sodium is going all one direction from the cytoplasm to the outside, and the potassium is going from the extracellular fluid being pumped in with the benefit of ATP being used, pumped into the cytoplasm. So a summarizing of the properties of cell membranes. It's a fluid-like phospholipid bilayer. Remember, there's a number of types of membrane lipids, but the majority are phospholipids. And the bilayer will also include cholesterol, proteins, and the glycolipids. They were all nicely depicted in that diagram that I pointed out in class today. Small molecules are going to get across by easy diffusion, simple diffusion through the bilayer. Things that are polar or ionic are going to require protein pores to get through the membrane, not just simple diffusion through the bilayer. So glucose and amino acids and some other kinds of things you don't have to know for sure are going to be transported with the aid of proteins not requiring energy. They're going to go from the area where there's more to where there's less. But the last one here is things that are ions that are going to end up with a concentration gradient across a membrane are going to require energy, and that would be active transport. So the last of our family of lipids is shown on this diagram that we began with at the start of the chapter, and the only one we haven't talked about yet is the eicosanoids. The name is inferring 20 carbons. That's what eicosa means. It's 20 carbons. And so the eicosanoids are all going to be 20 carbon molecules. They're derived from a 20-carbon unsaturated fatty acid or two, and one in particular is going to be more conspicuous in these examples. They tend to result as short-lived chemical messengers that are active near the point of synthesis. Their specific functions are not for your concern at the time, although you may have heard of some of these. There's two types that have specific names. Prostaglandins, leukotrienes are the two. You can imagine from the name that the prostaglandins were discovered in the prostate cells. They are conspicuously different from the leukotrienes because they have a five-sided ring in them. We'll see a diagram in a moment. And leukotrienes were discovered in leukocytes, therefore the name. Not that you have to know anything more of their functions for now. So one of them has a ring, one of them doesn't. And they all come from this one unusual looking unsaturated fatty acid called arachidonic acid. It's got 20 carbons and 4 double bonds. The result is it's got a crazy low melting point. And this arachidonic acid is converted into the different types of prostaglandins and leukotrienes. There are quite a few, several of each. Not that you're going to memorize arachidonic acid or its actual structure, but the idea is this. It's got 20 carbons, and there's a bunch of double bonds in there, and this unsaturated molecule, remember cis double bonds are common. The result is that you end up with something that bends around on itself, and this arachidonic acid is described as a precursor because it is formed into other things. A precursor does that, and it itself is actually derived from something else called linolenic acid, which is why we have that described as an essential fatty acid. Not that you're memorizing it, but one is a precursor to the next and so forth. But the precursor, then arachidonic acid, is made into other eicosanoids. Eicosanoids being the derivatives of arachidonic acid. They're ordinary lipids, and you can tell from all those carbons, well, 20 carbons and only one acid group, then this molecule is going to be quite lipid-like in its character, very much nonpolar, soluble only in nonpolar solvents. The diagram from the text on this slide shows you how if you bend around arachidonic acid and all the carbons are numbered, you can imagine where somebody's multiple steps that you don't have to follow are going to be able to turn it into this prostaglandin with notable five-sided ring in it, and yes, it's got some modifications, or modified into something else called a leukotriene, which doesn't have a five-sided ring in it, but yes, it has a bunch of other features on it. But in general, these molecules are quite nonpolar. They got a lot of carbons and not enough polar groups to allow them to be soluble in water. So they're lipids, and they have interesting features in terms of their properties or their characteristics, but you don't have to know a lot about them. Let's just mention a couple just to give you an idea. Prostaglandins have some therapeutic applications and a variety of biological effects, including things like stimulation of uterine contractions, and if anyone's ever been in labor, that makes you think. It has also to do, it's related to the antipyretic, in other words, fever-reducing effect of aspirin, because prostaglandin synthesis is inhibited by aspirin, and so aspirin is going to be able to block the effects of some of these prostaglandins. Aspirin also has to do with the synthesis of blocking the synthesis of an eicosanoid that is involved with platelet aggregation. So there's all kinds of interesting things. No, you're not memorizing or being tested on them. So the example with the prostaglandin and aspirin antipyretic effect is on this slide. If you take a look at aspirin, which you have done in lab already, acetyl group is on the salicylic acid, so that part on the right is the acetyl group, which reacts with an enzyme. We'll look at enzymes very soon, in fact next week, and that alcohol group of the enzyme is going to be replaced with the acetyl group from the aspirin, and that inactivates the enzyme by covalent change, and it inhibits the synthesis of prostaglandins when you have used aspirin in that way. So just to make sure you see it, there's the acetyl group, and it ends up on the enzyme in the long run. because of the nature of the way this process works. Interesting, but not something to memorize. The leukotrienes are involved in asthmatic responses. In fact, I think I've seen them mentioned in commercials on TV for allergy medications, but they're involved in asthmatic things, allergic reactions, inflammation, whatever. It's a lipid, it's an eicosanoid. More than that is not necessary to be a focus for you. So, here's some extra examples from the end of the chapter. You should be able to recognize what type of molecules these things are on the left and what type of process is involved. And those are leave for your consideration for the moment. You will note, I hope, that the molecule at the top is definitely a fat, and with the addition of potassium hydroxide, which is a base, you should have the ability to convert that molecule into, guess what, potassium salts of the fatty acids, namely some soap molecules, and glycerol is on the right. I'm not going to write it out. It's hard to do with this limited tool I have. So this molecule with the arrow right now is glycerol, 3 carbons, 3 alcohols. And the molecule below is somewhat similar, but it has more double bonds, quite a few more. And so being a triglyceride, it's got 3 esters on glycerol. You should be able to recognize this compound. It is going to be a triglyceride with a number of unsaturations. So instead of a fat, this compound would be an oil, most likely. Okay, good luck with the rest of your homework and good luck studying for the exam. Enough for today, and we'll see you on Monday.